good morning. First of all, thanks for the SMBE for this award. And, well, as it was mentioned, my name is Mariela, and I'm going to talk about the results of my doctoral research. The topic is, MAYEX is an ancient, long non-coding RNA recruited for X chromosome dosage compensation in a lizard. I worked with the lizard Anolis carolinensis. Its sex is determined by sex chromosomes, XX in females and XY in males. The XY system is very old. It was originated 160 million years ago, and its Y chromosome is highly degenerated. This means that the Y chromosome in males has only 7 genes left, having lost 98% of its genes. Both chromosomes differ in size and gene content, which is very different from what is seen in females, where they have 2 active X chromosomes. This creates an imbalanced gene expression levels, and this problem is also seen in placental mammals, marsupials, and Drosophila melanogaster. But all these species have mechanisms that help balance gene expression levels. For example, in placental mammals, one of the 2 X chromosomes in females is randomly inactivated. This mechanism is driven by the lncRNA 6. So 6 binds to several proteins, and one of them is the polycon group. And together, they mediate epigenetic changes to silence the transcription. On the other hand, in Drosophila melanogaster, the process occurs differently. Here, the single X chromosome in males is overexpressed to balance the gene expression between both sexes. Here, this mechanism is driven by the long non-coding RNA ROX2, and ROX2 binds to male-specific lethal complexes, and together they mediate epigenetic changes to active the transcription. So what happened in Anolis carolinensis? Well, in this project, first, we analyzed the RNA-seq data from embryonic tissues, with ranging from 5 to 15 days old. So we found a lncRNA that was expressing only in male tissues, and it was absent in females. Then, when we analyzed the adult tissues, we found that the same lncRNA was still present in males, but not in females. So this lncRNA is the only that was male-specific, so we name it MAYEX. Then we wanted to capture the chromosome conformation, so we used the HiC technique. In this technique, the nuclei were cross-linked, the DNA was digested, ends were ligated, and the library was sequenced. At the end, each interaction was aligned against the genome. In this plot, the blue dots are the interactions between 2 regions. So, when we analyzed the X chromosome contacts, we found a region where, surprisingly, MAYEX locus was located. Then, so MAYEX was having contacts along the entire genome. Then, into this region, MAYEX and repeats have a high frequency of long-range contacts along the X chromosome, up to 3 megabases. Then, we analyzed the topologically associated domains, so we found 6 stats into this X chromosome. Then, we'll look at the AB compartments, that A compartments are the active regions and the B compartments are the inactive regions. So we found that most of the X chromosome comprises active regions. After that, we used the technique, the Jarzig technique, because we wanted to look at the interactions between the RNA and the chromatin. So this is quite the same like high C, but at the end, each interaction was aligned against the transcriptome and the genome, respectively. So we found that not only MAYEX locus was having these interactions, but also the lncRNA MAYEX was having several interactions into this X chromosome. Then we analyzed the ChIP-seq data, and we found that the X chromosome is more acetylated than in males than in females, and the peak is and the signal is very even, except for a peak where, surprisingly, MAYEX locus was located. So then we compared the rich coverage of this epigenetic mark of MAYEX locus to the rest of the X and the autosomes. And we found that MAYEX locus has the highest levels of histone acetylation of the entire genome. With all these results, then we wanted to know what proteins are associated with MAYEX. So we performed a pull down of proteins, and it was absolutely amazing. Find a histone H4, an acyltransferase, several transcriptional activators, and structural proteins. So we suggest that MAYEX is involved in X chromosome activation. At this point, we needed to find a link between MAYEX expression and the regulation of the X chromosome. So we performed CRISPR-I experiments in analyzed lines. Here, I'm showing you in the control group the normal MAYEX expression, and the other represents the CRISPR-I experiment. So we found that in this experiment, we could downregulate MAYEX locus. So then we compare the gene expression of control group to each CRISPR-I experiment. So we measure this gene expression of 200 kilobases away from MAYEX locus. And we found that these genes were no longer expressed. So then we expand this region to 3.6 megabases. And we found that these genes also stop expressing. So MAYEX downregulation decreases expression levels of the X chromosome. Then finally we searched the origin and evolution of MAYEX. And we found that in birds, snakes, and agamids, next to the MORC2 gene, there's a lncRNA expressing in males and in females. But 160 million years ago, when the system shifted from CW to XY and a gene compensation emerged, this lncRNA that has expression in both sexes become male-specific, giving rise to MAYEX. So MAYEX is an ancient, long non-coding RNA recruited in males. In conclusion, we discovered a new mechanism of X chromosome dosage compensation in a lizard, and similar to what is seen in placental mammals and Drosophila melanogaster, it involves a long non-coding RNA spreading along the X chromosome and binding with proteins, working together to reshape the expression of the X chromosome. I want to thank everyone who was involved in all these amazing projects and the funding institutions, and I want to thank you for your attention.
We've got time for maybe one question.
Well, it's an amazing talk. I think it's a very interesting result, and what is great is to find this convergence. So, do you think other lineages might have evolved similar mechanisms that are still yet to be discovered?
Okay, so with the species that have dosage compensation, like I shown you previously, we know that it has it, but there's another lizard that doesn't have dosage compensation, so it could be maybe something different that is happening in all of them. So right now, we don't know.
Okay, thanks very much, Marietta.
Thank you.
Our next speaker is